Thank you so much for joining again for lecture number 4 of the BICD140 undergraduate immunology course. Okay, so in the past 2 lectures, we were learning about innate immunity. So today we will cover some aspect and introduction of adaptive immunity. And at the end, we will try to also cover the location of the immune system. So we will first look at what is clonal selection and expansion, the 2 major types of adaptive immune cells, how do they recognize pathogens and how do they work. We will talk about the dendritic cells that we introduced before as the link between the innate and adaptive immune system, and we will see how they do that. And we will also mention the interrelationship between innate and adaptive immunity. And as I said, finally, we will try to cover the location of the immune system. This is more of what we've been mentioning, and is that the adaptive response is specific to the current infection, to the infection in place. And so antibodies made during an infection with measles virus bind to the virus and prevent reinfection with measles virus, but will not protect you from infection what did I say? Antibodies made during infection with measles will not protect you for an infection with influenza infection, right? Because they are highly specific for example, the spike proteins, the proteins that are in the surface of the measles virion, and will not bind the proteins that are in the surface of influenza virion, right? And so they are highly specific. Although within viruses of the same family, there could be some cross-reactivity. And it has been proposed, for example, that T cells specific for SARS coronavirus 2 may be present in patients that never had been exposed to SARS coronavirus 2. So some people have pre-existing T cells that recognize SARS coronavirus 2 because they cross-react with other coronaviruses that have been around. And so again, they are highly specific for a virus, but sometimes can be cross-reactive because the sequence of amino acids recognized by the receptors of the adaptive immune system may be present in viruses from the same family. And for example, again, SARS coronavirus 2 have some sequences of amino acids in their proteins that are shared with other coronaviruses that have been around that caused a mild a cold and respiratory infection. Okay, they are really not pathogenic. Okay. So the adaptive immune response is based on the diversity of the receptors that are present in B cells and T cells, and then a clonal selection. So selecting the right receptor, right? The clone of cell, the cellular clone that contains the right receptors that recognize the infectious agent. And so there are billions of different specificities in our adaptive immune population. And so you can see this is depicted here. During development, progenitor cells in the fetus is giving rise to a large number of lymphocytes, each with different specificity. So this is the progenitor cell in the bone marrow. The common lymphoid progenitor, remember, is the one that originates and developed into T cells and B cells. And for each of the adaptive immune cells, for B cells and for T cells, you will have this scenario in which you will have a large number of lymphocytes that encode for a different receptor for the pathogen in their surface. So these receptors are similar, but they recognize different sequences in the pathogen, and it's an unlimited numbers of specificities. Okay, so even if we haven't been exposed to HIV, for example, we will have some very few, but some cells that probably recognize, are able to bind to sequences in the HIV proteins. Okay, but these are in very low numbers. So we have a large number of clones with specificities for a large number of substances potentially present in pathogens like in HIV, for example. But each of these clones is in very a low representation. Okay, so we have very few of each of the clones that contain the receptor that is specific for a particular sequence of a protein of HIV, for example. And so only those receptors that recognize the specific pathogen will respond during a particular infection. Okay, so in this pool that we have are born with, we will have receptors that recognize HIV, measles virus, influenza virus, but only those receptors that recognize measles virus will be selected when they encounter the pathogen, a protein for which they are specific. So, for example, the measles virus protein will be selected only the ones that recognize the infectious pathogen, and these ones will be expanded, which be clonally selected, which means they will be copied. Okay, this clone will be expanded, will proliferate and make many copies of this clone. And of course, we want to make many copies of the clone that has the receptor for the pathogen that is infecting us. That makes sense. So we want to have more of that clone that is the one that will help us, right? If we have a clone here that recognizes HIV, it will not help us if we are infected with measles. So we don't want to expand that HIV specific clone. We want to expand the clone with the receptor specific for the pathogen that is infecting us. And so we end up, we induce proliferation and differentiation of the pathogen specific lymphocytes to form clones that are not only increasing numbers, right? But also during this process of differentiation, they acquire the capacity to exert effector functions, and we will see what are these effector functions. Okay, and so, but these effector functions will allow now these clones that are larger in number and now are effectors because they have acquired the capacity to execute and perform these effector functions to eliminate the pathogen. Is that clear? I can make a pause now to answer questions because this concept can be a little bit confusing.
Yes, we had a question in the chat. They're asking.
Remember, you can. Thank you, Dina. You can unmute and ask the question. I'm super happy always to hear your voices and answer your questions.
The student's mic was a little broken, so I'm going to ask this one for them. Does the receptor have to have an exact match for the amino acid sequence to bind to the specific pathogen, or in this case where it's similar viruses, can the sequences be slightly off?
Let's say for this course they have to be matched or mostly matched. You know, they have to be matched. Let's say like that. There may be some exceptions, but for this course, I think that we need to remember that they need to be matched. So the sequences that for example, here, we may have expanded some of the clones that are specific for other coronaviruses we have encountered, and they recognize a sequence in the spike protein or other protein of these previous coronaviruses that are exactly the same as the sequences are conserved in the SARS coronavirus 2 that is causing COVID-19. And so this may give us some level of protection and pre-existing immunity that will make attenuate the infection with SARS coronavirus 2. And that's one of the explanations that scientists are coming up to explain the great variety of clinical outcomes that occurs in COVID-19 patients. Not the only one, but one of the explanations. Any other question?
I had a quick question. Regarding the specific, you said that we're born with a small number of adaptive lymphocytes. Where did they gain that specificity if, you know, the fetus isn't actually introduced to any pathogen?
Yes, excellent. You know how we can be born with a small number of lymphocytes specific for HIV that we have never seen. Ben, that's exactly the question you should be asking. So excellent. We will learn that on how these specificities are generated during development in the lecture 6 and 7. Okay, but for now, you need to know that we have many in our blood, right? We have many lymphocytes when we are born. However, so it's not that we have a small number of lymphocytes. We have many B lymphocytes and many T lymphocytes, but we have very few that are specific for a particular pathogen or a particular structure, okay? So we will have many lymphocytes, but we will have very few specific for HIV, very few specific for influenza. We can almost, it's very hard to detect even, they are so few, okay? Okay, and that's why it's necessary to clonally expand them. And that this clonal expansion of the pathogen-specific lymphocytes takes time. And this is why the adaptive immunity is slower. Okay, so we will learn in lecture 6 and 7 how this specificity, this great diversity in specificity of the receptors of the adaptive immune cells is generated during development. Yeah, so is that okay, Ben? Okay, yeah, any other question? Okay. So here I want to bring up also some vocabulary. So we are in the same page. And so these lymphocytes here that we have, before they encounter their a pathogen substance for which they are specific, they are called naive, okay? Or resting. They've never seen the ligand of their pathogen receptors, okay? And however, after they proliferate and differentiated to acquire their capacity to exert their function, for example, we'll see some of their function is to kill infected cells, they are called effectors, okay? And so at the time we see the pathogen, we only have naive lymphocytes in very low frequency of specificity for the pathogen. But after a week, we have many of the cells of the adaptive immune system specific for the pathogens that now are effectors because they have acquired the capacity to execute their functions, okay? So which one is true about adaptive immune system or clonal selections? Each lymphocyte bears a single type of receptor. Yeah, this is true. You know, we haven't covered everything, but this is definitely true. So each clone will have a receptor, will have many molecules, but all will be identical, and they will all be specific for the same ligand, okay? Potentially a ligand present in a pathogen. So interaction between a foreign molecule and a lymphocyte receptor capable of binding that molecule with high affinity leads to lymphocyte activation. Okay, this is the activation that allows the clonal expansion. So this is true as well. Okay, so this is depicted here. So interaction of the pathogen with a receptor with high affinity will lead to activation of this clone, followed by proliferation and differentiation into effector cell. Okay. C, so this is true as well. So C is the differentiated effector cells derived from an activated lymphocyte will bear receptors of identical specificity to those of the parental cell from which the lymphocyte was derived. And this is true. And again, this is depicted here. So the effector cells will share the same receptors that have the same specificity for the same ligand than the parental cell receptor that originated this effector cell, okay? So so C is true, and D says lymphocytes bearing receptors specific for ubiquitous self-molecules, and we haven't learned this at all, but this is true as well. So lymphocytes bearing receptors specific for ubiquitous self-molecules are deleted at an early stage of lymphocyte development and are therefore absent from the repertoire of mature lymphocytes. And so I put this so you start thinking that we are during development, there may be generated some receptors in the adaptive immune cells that recognize self-molecules, but we don't want to have those ones. So these ones will be deleted before they start circulating so they don't attack us, okay, causing autoimmune diseases. So they are deleted early stage in development and are therefore absent from the repertoire of mature lymphocytes that is depicted here, that will be circulating, as we will see, in lymphoid organs, blood, and lymph, surveying for finding the pathogen ligand. Okay, any question about that?
Professor,
I have a question.
Yes,
you
Emily.
Go ahead,
Oh,
Emily.
okay. I was wondering why we would generate lymphocytes with self-receptors at all, or like why they're created in development only to be deleted later on.
That's a good question. Yeah, that's an excellent question. I think that because the generation of these receptors, and we will learn more, is random. Okay, so because it's random, chances are that may recognize self-molecules. And so, and that's a disadvantage, but we can deal with that by deleting these clones that recognize self-molecules. But by doing it randomly, you also cover an unlimited number of specificities because you don't know what you may encounter in your life. And you may need to be protected for. We have a lymphocyte specific for viruses that are still not in, have not jumped into humans. It's an unlimited number of specificities.
Thank you.
Yes.
I'm sorry. It was Michaela.
What's another? Michaela.
Yeah. Hi. I did understand the difference between effector cells and naive cells, but I didn't jot it down when you said it. Is the difference just because the naive cells, they do have specificity, but they're also broad in the sense that there's a very few of them, depending on the pathogen that does come in. And then the effector cell is what proliferates and differentiates depending on which pathogen it is. Is that exactly what that is? Okay.
Yeah, you kind of got it. So the naive cells are there before. Naive cells is the clone before it has been engaged by the ligand of the receptor. Okay.
Oh, I see. Okay, that makes a
And
lot of sense.
once it is engaged by the ligand of the receptor, it will become activated. So it will not be naive anymore.
Okay.
And this activated clone will proliferate and over several days and also differentiate. So it will change the transcriptional program, acquiring the capacity to execute their function. And now it can be considered an effector. Okay.
Okay, thank you so much.
Sure. Any other question? Okay, so let's look at which are the major types of adaptive immune cells, and then we will look at how they recognize pathogens. So the effectors, the adaptive immune cells will learn that are B cells and T cells. So again, here are the naive, small resting lymphocytes. When their receptor is in the B cell or in the T cell, is engaged by the ligand that can be potentially present in a pathogen, it will become activated and it will increase the size, okay? This engagement of the receptors will induce a signaling, an intracellular signaling that starts making these blast cells, and it induce the entry in the cell cycle. And so it will become bigger, that is a sign that is in the cell cycle, and it will start proliferating, okay? And also will become differentiated, as we mentioned before, into effector cells that can execute their function. So for the B cells, the effector B cell is also named plasma cells. And it's basically an antibody factory. So it has enhanced tremendously the endoplasmic reticulum because it's specialized now at producing large amount of proteins, which are the antibodies. And we will learn where do they come from, these antibodies, okay? And basically, the antibodies is the same antigen receptor that the B cell has in the membrane, but now in a soluble form, and we'll learn about that. So the T cells, when they become effectors, they can not secrete their antigen receptor. So that's a distinction between effector B cells and effector T cells. But instead, the effector T cells, we will learn more about this, but will acquire capacity to kill infected cells, like the natural killer cells, but with other recognition receptors, and will also may also acquire capacity to activate other cells to kill the pathogen. For example, some T cells, we will learn, activate macrophages to enhance their microbicidal activity so they can kill the pathogen, okay? And this will become clear in the next few slides. So the effector response, or the immune response mediated by B cells, is also called humoral immune response. And so here you have this depicted, the humoral immune response. This is the microbe, in this case an extracellular bacteria. This is a B cell. Remember, we have many clones with unlimited specificities that are naive, but now some of these clones will recognize a part of this bacteria and with higher strength, and that will select that clone, will activate that clone, which will proliferate and differentiate into effector B cell. And the effector B cells, we said, are called also plasma cells that are like an antibodies factory. So now they will secrete their receptor that they had in their surface in a soluble form that is called antibody, okay? And because to begin with, this receptor recognize a part of this bacteria, now this antibody is exactly the same, will recognize exactly, it's the same protein, but now in a soluble form. So will recognize a part of the bacteria. And that we'll see how helps eliminate the bacteria. And so these antibodies are soluble, right? So are in the serum or the extracellular fluids. And so, and that's why they are called, because they are in these body fluids, they are called humoral immunity. So humoral immunity, or immunity mediated by B cells and antibodies, is named because it involves substances, antibodies, that are found in the humors, the body fluids. And so what about the T cells? It's called also cell-mediated immunity. And here we have an example for, let's see, for CD4 helper T cells. That is a subtype of T cells, CD4 helper T cells. And so this, when, for example, there is a macrophage that have phagocytose, a pathogen, sometimes the macrophage, for some pathogens, not very virulent, or no, they can exert their microbicidal activity and eliminate the pathogens. But some pathogens, like Mycobacterium tuberculosis, for example, they have evolved means to subvert the microbicidal activity of macrophages. So they cannot be eliminated from these vesicles in macrophages unless they receive help from these CD4 T cells. And so the naive CD4 T cells that have the receptor specific for this mycobacteria, for example, that is growing in the vesicles of the macrophages, we will see how it will be selected and clonally expanded, become effector CD4 T cells. And now that this effector can exert its function. Its function in this case is the activation of the macrophages, further enhancing their microbicidal activity that will lead to the death of this mycobacteria that is growing in the vesicles. So what about the other subtype of T cells, the CD8 T cells that are also called cytotoxic T cells? So they are specialized to kill, for example, virus-infected cells. And so here we have an example of a cell that is in which the virus is growing. This could be the endothelial cells or epithelial cells in our lungs, a respiratory tract, right? And this virus may be SARS coronavirus 2. And so the T cells that have their receptors specific for a little part of in a protein of this virus will be activated and will proliferate and will differentiate into effector CD8 T cells. And now that is effector, effector means that have acquired the capacity to execute the function. And in this case, the function is to lyse the infected cells. So now the effector cytotoxic CD8 T cells will be able to lyse the infected cells, putative and endothelial cell infected with SARS coronavirus 2. Oh, sorry, epithelial cells infected with SARS coronavirus 2. Okay, so, oops. Here is a cartoon showing exactly what I mentioned before. So here is the infected macrophage. In their vesicles, they have growing the Mycobacterium tuberculosis. And one of the mechanisms in which Mycobacterium evade the killing by the macrophages is because they impair the fusion of the lysosome with the phagolysosome. Some mycobacteria have evolved to do that. And so, however, these infected macrophages, and we will learn more about that, they are presenting some of the pathogen-derived products in the surface, okay? And so the T cells that recognize where receptors, and now they are effector, right? They are effector T cells that recognize with their receptors these pathogen ligands. now derived from the mycobacteria, they can now help these macrophages and allows the fusion of the lysosome with the phagosome and enable the killing of these mycobacteria. Okay, and here is the effector, the cartoon depicting how the effector cytotoxic CD8 T cells act. And so we have here an infected cells with the virus. Some of the viral proteins, particularly glycoproteins that are in the virion surface, are also expressed during in infected cells in the surface of the cell. And now the cytotoxic effector CD8 T cells also already acquire the capacity to kill because it's an effector cell, will recognize this pathogen glycoprotein and will be able to, or another protein, and will be able to kill the infected cell, okay? Okay, and so what we covered so far is that what is clonal selection of expansion is the proliferation and differentiation of pathogen-specific B cells and T cells that are also called lymphocytes. We learned that there are two major types of adaptive immune cells, the B cells that are responsible for humoral immunity or antibody-mediated immunity, and the T cells that are responsible for cellular immunity. In the case of helper CD4 T cells, they will help other cells like macrophages. In the case of CD8 T cells, will be cytotoxic when they acquire effector functions and they will kill an infected cell. So now, how do they recognize pathogens and how do they work? And so we will learn that the B cells recognize pathogens via BCR or B cell receptors, and the T cells recognize pathogens via TCR or T cell receptor. And so let's learn first how the B cell receptor works. So the B cell receptor as well as the T cell receptor is belongs to the immunoglobulin family of proteins because they have globular domains. And always the B cell receptor will have a heavy chain, here depicted in blue, and a light chain, okay? The heavy chain has a transmembrane domain in the B cell receptor, so this would be the membrane of the B cell, the plasma membrane of the B cell. And the BCR, or the B cell receptor, will have a transmembrane domain that is part of the heavy chain, okay? So it will have two heavy chains, is B cell receptor, and two light chains. And so the antigen binding site, and we will learn more about what is antigen, but it's basically the ligand for this B cell receptor, will bind in this region that is constituted, as depicted here, by the light chain and the heavy chain, okay? And so the light chain and the heavy chain, they have a variable region that is depicted here, is the upper part, okay? And this most distal from the transmembrane region, and a constant region that is the one that is at the bottom, that is more proximal to the transmembrane region. Okay, and so you can see here that the ligand or antigen binding site is in the variable region, okay? And this is called variable because it varies in many receptors because it's the one that determines the specificity. And we said that we have an unlimited number of specificity, so it varies in the different receptors that have different specificity, and that's how we combine many different ligands potentially present in pathogens. Okay, the difference between the B cell receptor and the antibody that is secreted by the plasma cell, the effector B cells, is that the antibodies lack the transmembrane. All the rest is identical, okay? The only difference is that the antibodies lack the transmembrane region, and lack the transmembrane region, and because of that, it can be secreted, okay, and is soluble. So this BCR is always bound to the B cells via the transmembrane region in the plasma membrane, and the antibody is always secreted, okay? Okay, now, if you think about it, the T cell receptor is like having two of these light changes, okay? So it has a variable region domain and a constant region domain. You know, it's the same as the light change. It has a variable domain and a constant domain, okay? And it has two of these change, okay? And but in this case, the two change will, it's called alpha chain and beta chain, and the antigen or ligand binding site will be determined or will be formed by a combination of the light chain and the beta chain, okay? And as the B cell receptor, it has a transmembrane region, and it always has a transmembrane region. This T cell receptor is never secreted, okay? So it only exists as a receptor associated to the cell, to the T cell, via the transmembrane region. Okay, what type of antigens do lymphocytes recognize? Or what part of the pathogen does the antigen receptor bind? Okay, the B cell receptor and the T cell receptor. Here, I want to introduce the vocabulary of what antigen means, and I mentioned it a little bit, but so we are in the very same page. Antigen is any structure that is recognized by an immunoglobulin BCR or antibody or the T cell receptor TCR, okay? And it's called its corresponding antigen. So we often say the BCR is specific for this antigen present in HIV, or the TCR is specific for the antigen present in the glycoprotein of coronavirus, okay? So, but it's any structure that binds and with a strong affinity to the BCR or the antibody, which is basically the same as the BCR, but except that the antibody is soluble, right? And or binds to the TCR. Okay, so now the antigens that are recognized by the B cells and T cells are a little bit different. So the B cells, like innate cells, they can recognize pathogens in their pathogen products or pathogen parts in their native form, okay? So this is the native protein, for example, and the antibodies or the BCR will bind to the native protein, okay? To the protein in their native form, okay? In contrast, the T cells, they only recognize the pathogen parts that once they have been processed and presented by antigen presenting cells. And this is depicted here. So this is the protein in their native form. It will be in the antigen presenting cells will be processed into small peptides, and these small peptides will be presented by the antigen presenting cells depicted here at the bottom in these molecules that are called major histocompatibility complex molecules, MHC molecules, and are very important because are the ones that the antigen presenting cells will use to present peptides derived from the protein antigen. So the T cell receptors bind to the complex formed by the peptide antigen and the MHC molecule. So that's the difference between how B cells or BCR or antibodies recognize the antigens and how T cells recognize the antigens. So T cells only see the antigens when presented in MHC molecules in the surface of antigen presenting cells. And so here is shown the two types of MHC molecules that we have. MHC class 1 molecule that will present antigens that are recognized by CD8 T cells. So CD8 T cells only see their antigens when presented in the context or by MHC class 1 molecules. And CD4 T cells only only see antigens when are presented in the context of MHC class 2 molecules, okay? So TCR binds to antigens-derived peptides bound or presented by MHC molecules that are major histocompatibility complex molecules. And so which are these professional antigen-presenting cells that are holding or displaying these MHC molecules to the T cells? Well, for the ones that express both MHC1 and MHC2 molecules are called these antigen-presenting cells. But MHC class 1 molecule is in every cell of our body, okay? So any cell in our body has MHC class 1 molecule and can present antigens to CD8 T cells, okay? But only professional antigen-presenting cells or antigen-presenting cells, what we call antigen-presenting cells, display MHC class 1 and MHC class 2 molecules so they can engage CD8 T cells and CD4 T cells. And so which are these antigen-presenting cells? Well, there are two that we have already talked about. Actually, all of them we have talked about, but two we mentioned in the innate immunity lecture. We said that macrophages also present antigen or are important for antigen presentation. And so macrophages is one of the antigen-presenting cells or APCs. And dendritic cells, we also said that they are very important antigen-presenting cells, and we'll learn more about that. And not only they have MHC molecules, but they also have constitutive expression of co-stimulatory signals that are important for activating the naive T cells and initiate the immune response, and we'll learn more about that. And so the antigen-presenting cells, or APCs, are listed here. We have dendritic cells, and we will see they are found in lymphoid organs, and we will learn more about where they are and how they traffic. They are in the tissues as well, in connective tissues and in the skin. The macrophages are everywhere in our body. So on all our tissues, we have macrophages. The monocytes are the circulating precursors of macrophages, and they can also differentiate into dendritic cells. So they are localized in the blood, but once they get into the tissues, they differentiate into macrophages or dendritic cells. And the B cells are part of the adaptive immune system and can present antigens that bind to their antibody receptors. And we have a slide to explain this in the next few minutes. And they can, and then we'll see where they are located in lymphoid organs and recirculate in our body, and we'll learn more at the end. So let's look at the dendritic cells, which are also called the most potent antigen-presenting cells, because they are key to activate the naive T cells. So these antigen-presenting cells, they can present antigens in class 2 and in class 1 and in class 2, okay? But only the dendritic cells are the ones that are professional and are essential to initiate the activation of naive T cells. And they are called a link between innate and adaptive immune system. And this is nicely depicted here. So the dendritic cells are in our connective tissues, and they express tons of pathogen recognition receptors that we learned in previous lectures, like toll-like receptor 4, for example, that recognize lipopolysaccharide. And so when the dendritic cells in the tissues see a bacteria or a pathogen that express the lipopolysaccharide, that is recognized via toll-like receptor 4, or again, any other pathogen recognition receptor that finds the pathogen ligand, will now make this immature dendritic cell become mature dendritic cell. And now this mature dendritic cell not only is presenting antigen in MHC1 and MHC2 molecules to the naive T cell, but it also upregulates expression of co-stimulatory molecules that are important to initiate the activation of this naive T cell. So it can differentiate and proliferate into an effector cell. And not only that, but we learn that this engagement of the pathogen recognition receptors with their ligands will initiate transcription of via IRF and NF-kappa beta transcription factors to induce production of cytokines and type 1 interferons, which are also important for determining the flavor of differentiation and qualities of the differentiation and the functional capability that this T cell, effector T cell, will acquire, okay? So they link innate and adaptive immunity because they first sense the pathogen with innate receptors, such as pathogen recognition receptors. And then they will uptake the pathogen and present antigens derived from this pathogen in MHC molecules that will be recognized by the TCR in the naive T cells. They also upregulate co-stimulatory molecules that will are important for activating these naive T cells. And they will also produce cytokines that will help the differentiation of these naive T cells into effector cells. And now these effector cells, they have acquired the capacity to, in this case, is a helper cell to help a macrophage. And so, for example, we'll go back to the tissue and we'll be able to activate a macrophage, so the macrophage can now eliminate, for example, a mycobacteria, okay? So I want to teach more now on which are the ways in which the B cells and T cells exert their function, okay, now in their effector phase, when they have acquired the capacity to exert their function. But before that, I want to make a pause to see if you have any question and that I could address. Okay. So let's move on, and maybe we can do some clicker questions at the end to see if you really understood. Okay, so the functional outcome of a B cell activation and the formation of an effector B cell is antibody production. And we just said that the effector B cell is a plasma cell, which is an antibody factory. And so these antibodies that will be secreted, because remember, antibodies is the soluble form of the BCR, they will be secreted, but then they will bind directly to the pathogen ligand or toxin ligand, and they will lead to neutralization of the pathogen as depicted here. For example, in this case, is the bacteria has produced a toxin that will bind to the receptor and will be deleterious for the cell of the host. The antibodies specific for this toxin can neutralize this toxin by binding to the toxic and then prevent it to bind to the receptor. And ultimately, this toxin will be ingested by the macrophages, because these antibodies, many antibodies have the capacity to opsonize, okay, enhance the phagocytosis by phagocytic cells such as macrophages, and ultimately this macrophage will degrade this toxin, okay? This is true with the viruses as well. And that's why a lot, without going too far, you have heard in the news, for example, that the president has been treated with antibodies against coronavirus. And these antibodies are directed to the spike proteins of the SARS coronavirus 2. And basically what they do is they neutralize the surface protein of SARS coronavirus 2, that is the one bind to the receptor in the cells that SARS coronavirus 2 infects. And so because the antibody directed to that protein will neutralize that protein, now the virus cannot bind to the receptor and cannot infect the cells. So it helps prevent infection by neutralizing the virions. So the antibody binding to the pathogen or toxin can also lead to opsonization. And this is depicted here. This is a bacteria in the extracellular space. And the antibody specific for this bacteria will opsonize this bacteria. And this is what we mentioned before. And this is because the phagocytic cells have receptors for the antibodies when they are accomplished to their ligand. And so this can help uptaking the bacteria or other pathogen and enhance the phagocytic activity of the macrophages or opsonize the pathogen. And finally, we already learned that antibodies can trigger the classical pathway of complement activation. And so this is a bacteria in the plasma. The bacteria will be recognized by antibodies. The antibodies can initiate complement activation, and we know that complement activation will lead to opsonization, but will also lead, so enhanced phagocytic activity, and also lead to lysis due to pore formation in the membrane of the bacteria. So in all cases, with neutralization, opsonization, and complement deposition, ultimately the antibodies will help the degradation of the pathogen or toxin by the macrophages. Okay, so this is how antibodies work, and this is the functional outcome of B cell activation, okay? So what is the functional outcome of T cell activation? So let's see the functional outcome of CD4 T cells. And in this case, here we have two different subsets of CD4 T cells, and we will learn more about this in the lectures after the first exam, right? So this is just an introduction. But here is depicted the functional outcome of this subset that is called T helper 1, okay? That is the one that helps macrophages. And so here the macrophage engulfs and degrades the bacterial producing peptides. So now these peptides derived from the bacteria will be loaded into the MHC class II molecules in the macrophages. Remember that macrophages are APCs, antigen presenting cells, that by definition they express MHC class I and MHC class II. And so with the MHC class II, they will the peptides derived from the bacteria or pathogen they have engulfed, and will present that in their membrane. And so this is the way they are telling the T cells, hey, I'm infected with this pathogen that has this sequence, this peptide. And so now the T cells that now are effector T cells, remember we are talking of T cells that have already undergone activation, proliferation, and differentiation into effector T cells. Now when they go into the tissues, they will have the TCR specific for the pathogen, and that's how they will recognize the peptide and the MHC class II molecule presented by the macrophage, and that's how they will know which cell they need to help, okay? And so they will release cytokines as well, that, but other types of cytokines like interferon gamma that will help activation of these macrophages and increase in the microbicidal activity so it can kill the bacteria. And also here in the bottom, we have a similar scenario, but in this case is with a subset of CD4 T cells called T follicular helper cells, TFH. We'll learn more about that. But this is a subset that is specialized to help B cells, okay? And so in this case, it's depicted how the BCR binds to the bacteria for which this B cell is specific. So it will bind to antigens in these bacteria that are recognized by the BCR in the surface of these B cells. And of course, they have many identical BCRs. This is a simplification. Rasaris will have many BCR molecules, but they will all be identical and they will all be specific for the same antigen, okay? So this B cell, each B cell will have a BCR specific for the BCRs specific for the same antigen, and they will recognize these bacteria. And after recognition, one of the outcome of this recognition is that the complex, the BCR and the bacteria are engulfed by the B cells. And now these bacteria will be again presented in MHC class II molecules in the surface of these B cells. And the CD4 T cells that, remember, have the TCR that recognize their specific antigen in MHC class II molecules, and these TFH CD4 T cells will now know which B cells they need to help, and will be the B cells presented their specific antigen in the context of MHC class II molecules. And so now this activated B cell will further differentiate into an effector cell, which is the plasma cell, and produce antibodies that will be the same as the BCR that they, in the first place, recognize the bacteria. And so the antibodies produced here will, because they are the same as this BCR, will recognize this same bacteria and help neutralize, opsonize, and activate complement when they bind to this bacteria. Okay. So what is the functional outcome, finally, of the other subpopulation of T cells, the cytotoxic CD8 T cells? And this is the activation, upon activation, they lyse infected cells. And so here we have a virally infected cell. And again, this could be endothelial cells, and this virus may be the SARS coronavirus 2. And so the virus that is replicated inside the infected cells will be producing and translating tons of viral proteins using the cell machinery. And so some of these viral proteins will be produced in the endoplasmic reticulum, and other ones will be in the cytosol, but we'll see how in later lectures, but there will be peptides from these proteins pumped into the endoplasmic reticulum, okay? And so these peptide fragments derived from the viral proteins that are pumped in the endoplasmic reticulum will be loaded into MHC class I molecules in the endoplasmic reticulum, and will then be presented in the plasma membrane of this cell where the virus is replicating and infecting. And so now the effector cytotoxic T cells, the one that have already been activated, proliferated, and differentiated into effector cell that has acquired the capacity to exert its function, in this case of kill, will go to the tissues, and in the tissues will be able to recognize the infected cell, because it will be able to see the peptides from the pathogen presented in MHC class I molecule, okay? So again, because any cell in our body express MHC class I molecule, not only cells of the immune system or not only antigen presenting cells, all cells in our body express MHC class I molecules. Any infected cell can be recognized by an effector CD8 T cells. Once the CD8 T cell is effector, can recognize any cell in our body that is presenting MHC class I molecules with their specific antigens. Okay, any question about that? Okay, so I want to go now into one slide depicting the interrelationship between innate and adaptive immunity. And so there is actually a unified concept between innate and adaptive immune system. So I want to put it all together in one slide. So the innate immunity will learn how via molecular pattern recognition, so the interaction between the PRRs and the pumps will lead to alarming and destruction of the pathogen. And that will slow the pathogen invasion, so we will slow the pathogen spread, and we learn, for example, how interferons create an antiviral stage and may diminish translation of viral proteins or or degradation of viral RNA to slow down the pathogen spread. It may also induce direct killing of the pathogen, and we learn how natural killer cells can do that. And they may also call for backup, so they will recruit other innate cells, and we learn how these cytokines, particularly chemokines, can recruit other innate cells, all towards slowing down the pathogen invasion. We just learned in the past few slides how the cells of the innate immune system, such as dendritic cells or macrophages, are able to present antigens, and particularly dendritic cells are essential to present antigens and initiate the adaptive immune responses, okay? That's why they are called the link between innate and adaptive immunity. And so the adaptive immunity now occurs with clonal recognition or clonal selection of T cells and B cells that have receptor specific for the foreign antigen that is present. And this will be followed, this recognition will be followed by clonal expansion or clonal selection, okay? And this will generate effector B cells that will produce antibodies, or effector T cells that will produce cytokines and chemokines as well, And cytokines like interferon gamma, I mentioned, that will empower innate immunity. So we learn how some CD4 T cells are empowering macrophages, for example, to enhance their microbicidal activity. And so many of the effector functions of the T cells, particularly CD4 T cells, are to empower innate immunity. But also T cells can directly kill, and we saw that for CD8 T cells. And finally, we also learned that the cells of the adaptive immune system, B cells and T cells, can develop long-term memory that protects us for reinfection. Okay. And so part of how the adaptive immune system works is by empowering innate immune system. Another example of how the adaptive immune system empowers innate immune system is what we learn about antibodies, right? We learn how antibodies ultimately lead to pathogen destruction by the macrophages, which are cells of the innate immune system. Okay. So in the last few minutes, I would like to cover where is our immune system, okay? The innate immune system we learn is in the tissues. We learn that macrophages are all throughout the body. For example, the dendritic cells are in the tissues, but also we have monocytes in the blood. And we have also innate cells in lymphoid organs, okay? And we will learn more about these lymphoid organs. The adaptive immune cells are also in lymphoid organs, especially when they are naive, the recirculating lymphoid organs, as we will learn. They are also in blood, in tissues, and they are also in lymph, okay? So they recirculate in the lymph. And just to be clear on what is the lymph, is the plasma that has leaked from the blood into the tissues, and so is now invading these tissues, and is the extracellular fluid, and is collected through the lymphatic vessels. And so all the lymphatic vessels will have valves that promote the lymph to go away from the tissues. And so when doing that, it's also collecting antigens that are present in the tissues that will be carried in the lymph, okay? And the lymphoid organs are organs that contain lymphocytes. That's what they are called, lymphoid organs. But they also contain other cell types that provides a structure and support the production, maintenance, and circulation of these lymphocytes, these T cells and B cells. And these lymphocytes are the primary cellular component of the lymph. And this is because we will learn how lymphocytes are recirculating in the lymph and the blood. But before learning that, I want to mention what are the types of lymphoid organs. And so we have primary lymphoid organs like the thymus and the bone marrow, where the lymphocytes are generated from these immature progenitors, such as the common lymphoid progenitors, and they become mature lymphocytes, okay? And after they become mature lymphocytes, they will recirculate in secondary lymphoid organs, okay? Where mature lymphocytes reside and recirculate, and an adaptive immune response can be initiated, okay? And we'll see how these secondary lymphoid organs represent the meeting place between the pathogen and the naive lymphocytes. So the immune response can be initiated. Always in these secondary lymphoid organs is where the immune response is initiated. And so the secondary lymphoid organs includes the spleen, the lymphoid nodes that are connected by the lymphatics where the lymph circulates, includes Peyer's patches in the small intestine and the large intestine, And we will see more details on that in the next few slides. So the lymphocytes continuously surveys the secondary lymphoid organs for evidence or in search of their specific antigens, okay? So as soon as the naive lymphocytes see the specific antigens, which could be a pathogen, it can be selected, clonally expanded, and become a effector lymphocyte that will now recognize and fight this pathogen. And so this is a cartoon here showing the lymphocyte recirculation. And so after lymphocytes are generated from primary lymphoid tissues like the thymus of the bone marrow, and this is all before the pathogen is encountered here on the right. And so you can see here is a lymphoid node, which is one of the secondary lymphoid organs. And so the lymphocytes, the naive B cells and T cells, will arrive to this lymphoid node via the blood and will leave these lymph nodes via the afferent lymphatics, okay? And may enter another lymph node here because we have many lymph nodes in our bodies. And so it may enter another lymph nodes via the efferent lymphatic and then again leave via the afferent lymphatic. And so we'll be searching for their specific antigen in all the lymph nodes in our body. And if it doesn't encounter any antigen, we'll continue searching in the next lymph node and next lymph node until the lymph will collect these lymphocytes via the efferent lymph that exit the lymph node via efferent lymphatic into the thoracic duct that end up in the subclavicular vein and gets into the blood. And this is how the lymphocyte now will get into blood circulation again and will start again getting into a lymph node via the blood, and then again leave the lymph node via the efferent lymphatic, and again may enter another lymph node and then leave and enter another lymph node, and so on and so forth. This will be the recirculation of a naive lymphocyte, okay? In the meantime, the lymph, we said, is carrying antigen from the tissue. And so if we have an infection, for example, in a peripheral tissue, the lymph will carry the antigens from this infection into the closer secondary lymphoid organ, okay? And so now the lymphocytes, the naive lymphocytes, are recirculating in these lymph nodes, and the lymph is carrying antigens to this lymph node. Okay, so this is ideal because now the lymph node will have the naive lymphocyte recirculating and the antigen derived from the pathogen. And so that's why the secondary, and I'm ending with this, the secondary lymphoid organ compartmentalizes the infection and provides a meeting place for the cells of the adaptive immune response.